Hi, everyone. So this is a presentation that was supposed to be given by my boss, Christophe, but he had some problem getting on the plane. So I will do my best. I had been working on it, so I hope I'm qualified to speak about it. So this is about the Metavisitor, which we had been working on in the lab for, I think that's 2 years now, more or less. This is a suite of tools and workflows for both detection and discovery of viruses from NGS datasets. So a super small introduction to viruses. So you probably all know that they can infect cells depending on what virus it is and what cell. So usually you have like this virus that enters the cell and then it produces some nucleic acid. Well, you have a number of other nucleic acids that may appear in response to that, so DNA, RNA. And that's the stuff that we can sequence, right? So why not use that to figure out what's present? And maybe we can even find some unsuspecting things in NGS datasets. So I'm going to start by showing the diagnosis of known viruses. So it's a Galaxy workflow. It's modular. It's very easy to change. So I think that may actually be one of the main points of Metavisitor, that it's quite easy to exchange certain modules. So we start with the reads. As in most analysis, we do the classical quality control, trimming of the reads, things that need to be done. This is especially important with small RNA, but okay. So we use one of the aligners here, and then sequentially we line them to things that we're not interested in, like the host genome, the phyx control that is very often in the sequencing lanes, and perhaps things that we already know. And then we're interested in the unmatched reads. So that usually, I mean, depending on what you have sequenced, but if you have sequenced like samples from a specific species, you have discarded already the majority of reads, and you can now focus on the interesting things if what you're interested in is contaminating reads or viral reads or however you want to call those. And then the next step is to assemble those reads. So whether or not you're using small or long reads, I mean, Velvet is quite good for really small reads, and then you can use Trinity or other assemblers for longer reads. But again, I mean, as this is a Galaxy workflow, it's quite easy to change that. So then we assemble that, we get out the longer contigs, and we can use those to blast them against a database of known viral DNA sequences. So we have this query string with which we have a tool that can take query strings to the NCBI. And with, for example, the query string that is shown here, we can retrieve all viruses and exclude the cellular organisms. So once we have those, we make a blast database, and we can blast our assembled contigs against the database. And then we have this custom tool, in addition, that takes the blast outputs and makes a nicely formatted table. So I'm going to show you one example that has one of the examples that we have recently published in collaboration. So there is, this is the whole workflow. So we're downloading sequencing data sets from the SRA. We do as I said, the clipping, all the steps down here, they are relatively standard. One thing that we do is we collapse all the sequence. It may seem counterintuitive, but we find that the discovery diagnosis works just as well when you collapse all the sequences that are identical, which with small RNAs, even more of a reduction. And then, as I said, we align it to the host genome or other things that we're not interested in. And then we run Oasis Optimizer, which just takes Velvet with different k-mers. Yeah, and then the output of that phase is this table here, where you see the different hits that BLAST has found, and then how long those hits are and how much of that is covered by the config, and so on. And yeah, I mean, we can also take then the assembled context and the hits, go back and fill that in with sequences from BLAST so that you get like this context. They are quite often not complete. That depends on the characteristics of how the viral derived, how the reads are derived from the virus, whether it's the genome or in the case of small RNAs, they're very often degradation or products of cleavage. And we can fill those in, for example, with Cap3. Yep. So we can rediscover the stuff that is already there. And then the final output is the reference sequence. I mean, the new sequence that we have found, and the parts that had not been assembled through the workflow are on the top in the lowercase. So in that particular case, we were able to assemble the whole genome, and just the very beginning we had to take from filling in with the closest reference. So you can do that for all the hits that you have in your sample, and that gives you a nice overview of what is there. Yeah, there is another tool that is, I don't think you can really read that, but it's, okay, so at that, I mean, I was saying that you get this table, so you select the genome that you want, and then you go on and you just assemble that specific part. So it's really quite easy. I mean, we have developed this together with biologists, and we have deposited a preprint for this workflow. So it's on bioRxiv, and we have submitted it to +1, I think. And there is a proof of concept on a study that had recently profiled cells from HIV patients. We had cells from, we had sequencing of Ebola and Lassa virus, and we could also take just patient data, and we found like the infection with, we found, depending on the sample, infections with dengue, Hep C, and other viruses. So, yeah, I mean, it's there. And we can also find new viruses or potentially new viruses. So instead of blasting against the DNA at that step, we can also use a blast X step to blast against proteins. So that gives you more distantly related hits. And you can do that for the, you can look at those that had not been identified with blast N, and then go a bit further in the distance. And again, you can use the same steps of the workflow to get your new sequence back. You can fill it in, and you can use that to design primers and to confirm that what you found is really present and not just an assembly artifact. And this is a collaboration that we had been doing the assembly and the development of the workflow, and the guys there had been using that on Anopheles gambiae, and they actually, using this, were able to confirm 2 new viruses in their samples. So the complete workflow, the complete suite is available. We took like good care that this is completely accessible and reproducible, and that people can confirm it or use it with their own data. So it's present on our lab server. This is the Mississippi server. It's available in the toolshed. We have for our lab a repository of tools that is modeled exactly after the IUC repo. Again, this is for the tools that you need to run those workflows. The workflows themselves, it's the same thing. They are available. They're also on my experiment. And I already mentioned we submitted it to +1, and the preprint is on bioRxiv. And last but not least, we also have a README page on Read the Docs that specifically tells you how you can use it, how you can start your own instance, and what steps you have to take. So if you're interested by this, I encourage you to try it. Yeah. So that's it. I think I went through it a little bit too quick, but I guess that's never really a problem. So it was mostly, it was done in collaboration with biologists from the Institut Pasteur. It was a project that Christophe, my boss, started some time ago. This is a student that had worked on it to finalize it just this summer. And I guess I was mostly in the background, making sure that our servers run and, just reviewing the tools. So, one thing that we're still trying to do and trying to improve is the references, because we are downloading the references from the NCBI, and if you just ask for virus sequences, you get like a whole lot of things that you're not interested in if you're really interested in the biology of viruses, because you are in identifying what sequences are there. The set is very, very big, so if you take that as a FASTA, you line it, you get like 50,000, 60,000 or much more sequences, so that's a problem for the metadata. Actually the student, Juliana, she's working on trying to reduce those references. And I think this can be adapted to things that are outside of the virus, so outside of the viral field. We can think about making sure, looking at how many reads we have actually found and see whether this is different across different samples. So, we might go to bacterial metagenomics, but I see that there is already quite a few good things out there that have been also presented yesterday and in the training. So, thank you.
Any questions for the speaker? Going once, going twice. So I was looking at the workflow that you had put up there, and it looked like you were mostly pulling RNA-seq data, because I saw the Trinity and Oasis step. So how is that collected? Kind of echoes too, so it's a little weird. I was asking about the workflow that you showed. You were mapping it against known references first and also doing a de novo step.
Yeah.
So I was wondering how that relates to the raw data you were collecting. Were you collecting RNA?
So the experiment where it was published in +1, it was small RNA, yes. So for small RNA, I mean, they're really small. If you just blast them, you get like this whole lot of hits, but you can make it more specific by assembling them. That was the idea. So, and then if you're already working with longer molecules, like if you have done RNA-seq with standard length, then it might be more efficient to use assembler that work better with longer reads, like Trinity. That was the idea, yeah. But it also works with DNA. I mean, whatever type of sequencing you have. RNA is interesting for viruses because it correlates with a viral load. So.
Great, one more round of applause, please.